This is the second video in our series about parts of the cell, looking at the different organelles in eukaryotic organisms. We'll also see a little bit about the ancestors of the particular organelles that we are talking about in this video. We're looking at the energy organelles, the energy molecules that are important for these organelles to function. Couple of important words that we need to get started with. We may have seen these before. Now you definitely need to know them and be able to define them. Metabolism is the series of chemical reactions that will occur within cells, often moderated by enzymes. I have a slide coming up later that gives a little more detail of what enzymes are, but they are proteins that enable chemical reactions to happen. That's the most important thing that you need to know. And these proteins have very special shapes. They have very special shapes because of the order of their amino acids, which means we have hydrophobic sections, hydrophilic sections. That causes this protein to twist and turn in particular ways and do certain types of work, which proteins are very good at. Most of the work within the cell is done by proteins. Anabolism is when you build things up. You are doing dehydration and synthesis. You're taking amino acids, you're making proteins, you're taking fatty acids and glycerol, and you're making triglycerides. You're taking glucose or some other monosaccharide. You're making a polysaccharide like amylose or like glycogen or like cellulose. Catabolism is when you're breaking apart big molecules into smaller ones. Hydrolysis is the process where you take a polymer and you break it down into monomers, removing water in the process. Hydrolytic enzymes are the ones that make catabolism happen. Hydrolytic enzymes do hydrolysis. They take an enzyme, they are an enzyme that will break a monomer, such as a polysaccharide, maybe amylose, maybe a protein. They break it down into amino acids in the case of a protein, or into monosaccharides in the case of amylose, or cellulose, which certain bacteria have the ability to break down. Metabolism, metabolic, that's the describing word, that's the adjective. Pathways are the metabolic pathways are the chemical reactions that will occur in a stepwise process. Life has to do things very carefully. Life cannot be doing things randomly, as often happens in nature. Life needs to organize processes very, very carefully in order to ensure that energy release isn't too high, raising the temperature of the cell, or making harmful waste products, which is also dangerous to the cell. Life must control these chemical reactions very carefully, and it has set out certain metabolic pathways in order to make that happen. The chemical reactions are in a stepwise process. As I said, I just wanted to highlight this word: biomolecule, life molecule. Macromolecule isn't necessarily alive. Biomolecules are definitely necessary for life. What we know about mitochondria and chloroplasts is that at one point in their existence, they had been bacteria, free-living bacteria. Mitochondria had the ability to use oxygen in order to drive the production of energy in the cell. And that was a huge advantage when a larger eukaryotic cell ended up taking in the mitochondria. We see evidence of mitochondria being once independent bacteria because they have a circular DNA chromosome, just like bacteria. I will add that chloroplasts also have a circular DNA chromosome, which a different kind of bacteria, the cyanobacteria, have. If you notice, when this eukaryote incorporates the cell, the mitochondria, there's one membrane for the one cell membrane for the mitochondrion, or the bacteria that was originally there. There's another cell membrane that the eukaryotic cell used to surround the bacterium. And this double membrane structure is good evidence that at one time the eukaryotic cell took in a bacteria that turned out to be useful to make energy from oxygen. And this bacterium ended up surviving in the cell, became the mitochondrion, or in the plural, mitochondria. This is another good evidence that you have this model of endosymbiosis. Endo meaning go in. So this is endocytosis originally. If you remember our last video, endocytosis is when the cell brings something into it. Cytosis is when the cell is doing a process. Symbiosis in this case means two things working together. That's a very important vocabulary word, symbiosis. When organisms work together, we call it symbiosis. They work together for their own survival. In this case, the mitochondrion would have gotten protection from the eukaryotic cell, and the eukaryotic cell would have gotten energy from the mitochondrion. As we looked at the mitochondrion, we also have the chloroplast, which at one point was a cyanobacterium, a certain type of bacteria that does photosynthesis. The process, as we said, when chloroplasts came into the cell originally from a independent living cyanobacterium, we call that endosymbiosis, the same as the mitochondrion. Chloroplasts, if you remember, bacteria have a peptidoglycan layer in their cell wall. Chloroplasts, at least some of them, also have a peptidoglycan layer in their cell wall, which is good evidence that chloroplasts once were independently living cyanobacteria. Now, a little bit more about enzymes. A couple of vocabulary words we must understand. The substrate is some sort of molecule. In this case, we have a sucrose molecule. A reaction needs to happen with this sucrose. In this case, sucrose is the substrate. If this were a protease, which is an enzyme that breaks apart proteins, the protein would be the substrate, and it would break apart the protein into amino acids. It could be a carbohydrate. Whatever the substrate is the molecule that an enzyme needs to act on. It's a chemical that needs to react in some way in order to make a useful reaction within a living organism. In this particular case, this is hydrolysis going on. We see the water molecule going in, breaks apart the sucrose into a glucose and a fructose. This is the sweet table sugar that you're probably used to eating. So enzymes are proteins that catalyze. Catalyze means they make a reaction happen, usually at a lower energy level than normally would occur. If we were to break apart sucrose into fructose and glucose, it would usually have to happen at a fairly high temperature, too high for living organisms to tolerate. Enzymes allow this process to happen at lower temperatures. Substrates can react at lower temperatures so that life around this can still survive. The other thing you need to know, another vocabulary word, the active site. This is where an enzyme will bind to its substrate and allow the chemical reaction to occur. You can notice here how the sucrose, wait for the sucrose to come again, the sucrose will travel, has to go into this active site, the water has to come in, and then this is what makes possible the reaction of hydrolysis to occur. When we talk about energy in the cell, we're primarily talking about ATP, adenosine triphosphate. As you can see, we have carbons at these locations where there is no atom identified, nitrogens, carbon, nitrogen, we have oxygens, we have phosphates, carbon, hydrogen, oxygen, nitrogen, phosphorus is a nucleotide. This is a nucleotide. That's the macromolecule we're talking about here. So nucleotides can be used in energy storage. Most cell processes use ATP in order to work. This ATP, adenosine triphosphate, 3 phosphate groups. That last phosphate has a high energy bond. In order to get energy, the cell will break this bond, as you can see here. It will break this last phosphate in order to get energy. It will turn ATP into ADP, which is a low energy molecule. Adenosine diphosphate is ADP, as we see here. ADP is low energy. In order to charge it up to give higher energy to ADP, you need to add on that phosphate. When this process happens, it's an endothermic reaction. This absorbs energy. That energy is then stored in that third phosphate bond. When this ATP is turned into ADP, energy is released. That's an exothermic reaction, and therefore energy is released. And that is how the cell stores energy and gets energy it needs for all sorts of reactions that happen within the cell. The process of cellular respiration is how glucose, which is an important molecule in cells, is broken down in order to make ATP. As glucose is broken apart, energy is released. That energy is released and is absorbed by ATP. In this case, the first step is glycolysis. Big word, easy to understand. Glyco refers to glucose. Lysis always means breaking. Glycolysis is breaking glucose. That's the process. I want you to understand some of the vocabulary here. It'll be helpful. Lysis. Every time we see lysis, we're breaking something apart. Hydrolysis, we're breaking apart water. Cytolysis, we're breaking apart cells. Glycolysis, we're breaking apart glucose. Glycolysis occurs in the cytoplasm, not in the mitochondria. You may have heard the mitochondrion is the powerhouse of the cell. That is true, but this process of breaking down glucose happens in the cytoplasm, does not happen in the mitochondria. It requires 10 different enzymes, each of them doing different steps. It produces, out of a 6-carbon glucose, it produces two different 3-carbon molecules. Those 3-carbon molecules are called pyruvate. It also produces NADH, which we'll talk about a little bit, and ATP, which are those energy molecules. You will not have to memorize nicotinamide adenine dinucleotide, NADH. NADH is fine, but that is an important molecule for this process, and that is something that actually is necessary for other parts of this cellular respiration to occur. As I said, NADH is a cofactor. Cofactors help enzymes. In this particular case, it helps the enzyme move electrons for chemical reactions. Most chemical reactions, as you will also learn in chemistry, most chemical reactions do require movement of electrons from one atom to another. That causes the atoms to release energy or absorb energy, which is really how most things occur, how energy is released and how energy is absorbed. Glycolysis is independent of oxygen, does not require oxygen. You may have heard me say that prior to about 2.4 billion years ago, there was no free oxygen in the atmosphere or in the water. So this process, glycolysis, probably happened before the abundance or the presence of oxygen in the atmosphere. We call O2 free oxygen. That's the oxygen in its elemental state. Before this time, oxygen was bound up in other compounds like CO2 and H2O. Oxygen, O2 was not available. What we think is that actually free oxygen originally was very dangerous to the complex molecules that make up life. Now, most life did find ways of adapting, or many forms of life did find ways of adapting. But at the original start of life, free oxygen probably was not in the atmosphere, and it needed to be an evolutionary process where oxygen started being created and organisms needed to adapt to the presence of oxygen. And now we absolutely need oxygen. A little bit later on, we'll see why oxygen is so important in terms of making energy for a cell. Independent of oxygen is an anaerobic pathway. Anaerobic means no oxygen. Aerobic means oxygen. Think of doing aerobics, exercise. You're breathing heavy. That is oxygen requiring. Anaerobic is when maybe you're sprinting. If you're sprinting, you're moving too fast for oxygen to get to your muscles. So what your body does is produce lactic acid instead of breaking down to carbon dioxide. Lactic acid is a chemical that can be in your cells. It will make your cells burn, and then it will make your cells sore, which is why this process is necessary for reactions to happen. Just for the record, sorry, I got distracted for one moment. But glycolysis is anaerobic. It does not require oxygen. Lactic acid fermentation is something that happens within our muscle cells when we're moving too fast for oxygen to get to our cells. Lactic acid is what causes the burn in your muscles. It makes your muscles sore afterwards. Another very important process would be ethanol fermentation, which yeast, which are a type of fungus, is able to do. Ethanol is what we also call alcohol. It's the chemical that's in beer and wine that is necessary for this poison that we call beer or wine or alcohol. Ethanol is a poison like every other alcohol is a poison. If you're an adult human, you can break down ethanol in small amounts, but you can get ethanol poisoning if you drink too much, as unfortunately happens a lot of times in colleges. So drinking is not necessarily a great thing to do, definitely not when your brain is developing. But even if you are an adult and your brain is fully developed, ethanol can still cause chronic long-term poisoning. It's kind of a dangerous thing. We want to avoid drinking too much ethanol at any point. Ethanol is produced in an anaerobic process. Yeast, these fungi, if they're given the opportunity to break down sugar, they can make their byproduct alcohol, carbon dioxide, and energy, although this process is not very efficient. However, people do this in order to produce ethanol. That was step 1 of cellular respiration. Step 2 is called the Krebs Cycle, also called the citric acid cycle. You don't have to worry too much about all of the steps, but you do have to know that this occurs within the mitochondria. Very important to be aware of that. Pyruvate, that 3-carbon molecule that is a product of glycolysis, enters the matrix, the mitochondrial matrix. This is the inside of the mitochondrion. This is what was originally the inside of the bacterial cell that mitochondria are made of. So this bacterium had the circular chromosome of DNA. That's where this DNA is still found within the mitochondrion. The pyruvate, this 3-carbon molecule, moves into the matrix. This process requires oxygen. It is oxygen dependent. It was named after Hans Adolf Krebs, who was English, he was born in Germany, but he was English. He identified this pathway along with William Arthur Johnson. Krebs was the one who won the Nobel Prize in Medicine for it. There are 8 different steps to the Krebs cycle. You do not need to memorize them, although if you do go on to AP Biology someday, you may need to memorize them in AP Bio. Each of them is catalyzed. Remember, catalyzing is when you take different substrates, bring them together for the reaction. It is catalyzed by several different enzymes in these 8 steps. And enzymes are the proteins that make chemical reactions happen. What we produce from the Krebs cycle is NADH, this important cofactor, this important molecule that helps helps enzymes move around electrons. GTP is pretty much the same thing as ATP. GDP is another high energy molecule. FADH2, which has 2 hydrogens, or 1 extra hydrogen from FADH. FADH2 is another cofactor. What this Krebs cycle does is it produces NADH, that important cofactor. We saw that in glycolysis. GTP, which is an energy molecule just like ATP. FADH, which is another cofactor. It's going to be a source of hydrogen ions, as we'll see very soon, and electrons. And it also produces carbon dioxide. This is the thing that we breathe out when the carbons are broken from the glucose-derived molecules, which are the pyruvates, the 3-carbon pyruvates. When those carbons are broken off and form CO2, energy is released, which goes to GTP and also the hydrogen atoms and electrons go to these molecules. One turn of the cycle will make 2 CO2, so 2 carbon dioxide from that 3 carbon molecule, 3 NADH, 1 FADH2, 1 GTP. Do you need to memorize all of that? No, but just know that one glucose molecule has 6 carbons. Pyruvate has 3 carbons, and therefore you make 2 pyruvates from 1 glucose. And these pyruvates will then go through the the Krebs cycle. And this Krebs cycle will then require 2 turns, each starting with 1 3-carbon pyruvate. It will require 2 turns for a glucose molecule to be broken down completely. And in this case, we're making CO2, we're making NADH, which has an extra hydrogen on it, which will be very important very soon. FADH has 1 extra hydrogen on it from FADH, which is the original molecule. And then we have GTP, which is an energy molecule, just like ATP. The last step in this process is oxidative phosphorylation. Big words. Phosphorylation. We're making phosphate. We're joining ADP. We're joining an extra phosphate to make ATP, just to be aware. This happens within the bacterium. This is step number 3. We're taking hydrogen ions and electrons from that NADH. NADH will become NAD+, because we're taking a hydrogen and an electron from it. FADH2 will become FADH, my mistake. Let me add that in right here. FADH. And FADH loses 1 hydrogen and 1 electron, 1 hydrogen ion and 1 electron. These hydrogen ions are pumped outside of the intermitochondrial matrix. They are passed along to oxygens. And really, all you need to worry about here is there's something called the electron transport chain. Electrons are moved one way, hydrogen ions are moved the other way. We establish a concentration gradient where these hydrogen ions want to come back into the mitochondrial matrix instead of being out here. This is what diffusion wants to happen here. What happens is you have this very important enzyme called ATP synthetase, which allows the hydrogen ions to come back through. In that process, it actually takes ADP and makes ATP. Very interesting process. We push out the hydrogen ions, right? Now, this was supposed to be animated, but it actually turned out not to be. The mitochondria's inner membrane is the barrier. Hydrogen ions are on the outside. Hydrogen ions want to come inside, and in that process of them moving, the mitochondrion is able to capture energy and take ADP and turn it into ATP. This is 15 times more ATP is produced in this process than glycolysis alone. The overall reaction we can see here, you take 1 glucose, you take 6 oxygens, you make 6 carbon dioxides and 6 waters. This is aerobic respiration because oxygen is involved. The energy in 1 glucose is transformed into the energy is captured by 38 ATP molecules. If we're able to do this anaerobically without oxygen, it would be much less efficient. Each glucose that we break down, we would only generate a total of 2 ATPs, which still is something. It's still useful in some way, but not nearly as efficient as generating 38 ATPs. Now, with this video, I will stop it right now before we get to photosynthesis, which is the next reaction that we want to look at.